All right, we're going to introduce the topic of DNA. You guys have already read 2 sections about it. So we're going to go over the structure of DNA. We're also going to go over and discuss the individuals who contributed to our knowledge about DNA. The scientists that we're going to go over, they all were trying to figure out what was the source of the genetic material. And through their experimentations, they found out that it was DNA and not other things. So our current knowledge of DNA is only about 90 years old. We learn more and more information each year about DNA. First discovered in 1928 or around that time. And then it says that they were able to actually map all of our genes. For example, in humans, we have about 30,000 genes that they figured out in the early 2000s doing something called the Human Genome Project. And again, like I mentioned before, there are tons of new discoveries being discovered all the time about DNA. So the first scientist that we're going to talk about is Frederick Griffith. And what Frederick Griffith did was he studied pneumonia. Pneumonia is caused by bacteria. That is why when you have pneumonia, they give you some sort of antibiotics along with other things, because the antibiotics work with bacteria, but they do not work with viruses. So one of these forms of bacteria is harmless. The other one is harmful. So the pathogenic, anything that causes disease is termed pathogenic, and anything that does not cause disease or is harmless is non-pathogenic. So he injected mice with the harmless form. Nothing happened to the mice. He injected the harmful form, and obviously the mice died. He then injected a combination of the harmless and the harmful bacteria, and he noticed that the bacteria died. He was wondering why. Well, bacteria have a process called transformation. They can take up the genes or the genetics of another bacterium and make it part of their own. So the harmless bacteria took up the DNA of the harmful bacteria, and that's why when they were together, the mouse died. Okay. This is just pictures of what we were talking about there. You have similar pictures in your textbook showing us the different situations about injecting the mice with harmless and the harmful forms, and then the transformation process, which is illustrated right here. All right, so the process by which bacteria are able to take up the genetics of other bacteria and implement it into their own is called transformation. Now, the factors that they're talking about are genes or traits or whatever it is. All right, so Oswald Avery. Oswald Avery is another scientist who piggybacked on the other experiment with dealing with mice, okay? The transformation process. So Oswald Avery also worked with heat-killed bacteria. And again, they were trying to figure out what was the transforming principle. Was it a protein? Was it carbohydrates? None of that caused a change in the organisms like nucleic acids did. Okay, so it was the nucleic acids, DNA in particular, that were passed on and caused a change in the transformation principle. All right, Hershey and Chase, what Hershey and Chase did is that they discovered that if they use radioactive material, now the radioactive material is important because they can actually use this to trace through an organism, through the body of an organism, maybe where the item is that they're looking for. So what they did was that they used radioactive phosphorus to find the DNA and radioactive sulfur to find the protein. And when they allowed these bacteria to reproduce, the bacteriophages, which are viruses that infect bacteria, when they allowed the bacteriophage to reproduce, they were going to see what was passed on to the new organisms. Well, what was passed on is the DNA. Okay, the radioactive DNA was passed on to the new organism and not the phosphorus. Thus, supported their principle that DNA is the source of the genetic information. So these bacteriophages, they look like either an alien spaceship or some kind of alien. Now, here is the phosphorus, which is found in DNA. Okay, so as we see, when the DNA was injected inside, this was what was passed on. The protein coat was not passed on. Again, that supported their experiment and suggested that DNA is the source of the genetic information, not the protein coat. We know that from previous lectures, we know that all of our heritable information, the stuff that we pass on, is found in our genes. We also know that through the process of mitosis and meiosis that our genetic information is copied fairly easily. It's not a very detailed process. And we know that these characteristics, these heritable characteristics, are passed on from parent to offspring along generations. So there are three parts to the DNA molecule, okay? Our DNA's main part, building block, is a nucleotide. A nucleotide has three parts. It has a 5-carbon sugar, has a phosphorus group, or phosphate group, which is made of phosphorus, and then it has one of 4 nitrogenous bases. Adenine, guanine, cytosine, or thymine are the 4 bases that we find in DNA. So here are the 4 different bases structurally. The 2 smaller ones are cytosine and thymine. They're called the pyrimidines. We can tell that they're smaller because they only have 1 ring that represents their structure, whereby the purines are double. They're larger. They have 2 rings that represent their structure. All right, guys, so our 4 different bases here, cytosine, thymine, obviously are the smaller ones. How do we know that? Because they are single ring structures. They're called pyrimidines. And adenine and guanine are the larger ones. They are called purines. And we can recognize a purine by looking at the double structure there. This Erwin Chargaff. What he came up with is called Chargaff's rules. So we have 4 different organisms here. Now what you're looking at is look at the approximate amounts of adenine compared to thymine, and then guanine with cytosine. They are not equal, they are approximately equal. So Streptococcus has about 29.8% of adenine and 31.6% of thymine. That is not equal, but it's approximately the same. So, all right, now this scientist is the individual who discovered the process of X-ray diffraction. She also discovered that there were 2 forms of DNA, an A form and a B form. Now, interestingly, she is the reason why Watson and Crick were able to build their model. They finessed her model. No, yes, they did. They stole her picture, her X-ray picture, and they used that information without her knowledge to build their model. Now, ultimately, what they got was that they got a Nobel Prize from building this model. Rosalind Franklin, due to some complications, she got cancer from probably dealing with a lot of the X-ray information. She died. She did not get any Nobel Prize whatsoever. They get most of the credit, yes, and it was because she was a woman, and at that time, women were discriminated against. And the other thing is that when they were working in the labs and stuff together, her lab office was down in the basement. Their lab office was up on the floor somewhere. So it was not a very hospitable place for a woman scientist to be in. Here's a picture of her X-ray diffraction photo. And again, this is the information. Now, we look at it like, oh, okay, well, we can't really tell. We see that some X's here, but it's actually a double helix. Is a double helix, and people who study X-rays are able to see that that is a double helix. So Watson and Crick were able to take this information, and they were able to build their model. All right, so here are the some pictures of these individuals. We have Crick, Watson, who's an American, Maurice Wilkins, he was the director of the lab that they all worked in, and there's Rosalind Franklin. Now it says that they made cardboard cutouts of the nucleotides. Well, they did not know how to place these nucleotides and what order to place them until, again, they saw her X-ray diffraction photo. Then it was an aha moment, and they were able to figure out that DNA runs from 3' to 5' on one end and 5' to 3' on the other, which means that it's anti-parallel or it runs upside down. Picture of Watson and Crick. and this is the model that they made out of metal parts and things like that. But they are very famous for creating this model, which gives us a basic understanding of how DNA looks and works and operates. I told you that DNA runs in opposite directions. So one side is going from top to bottom, and then the other side is going from bottom to top. Now the 5' represents where the carbon is. So that's the 5th carbon. 1, 2, 3, 4, 5. And the 3' right here is the 3rd carbon. So that's 1, 2, 3. All right, so we have to recall and remember talking about how the letters complement one another, or base pairing in DNA. Adenine complements thymine. It goes in the opposite direction as well. So thymine is going to complement adenine. Guanine complements cytosine, and cytosine complements guanine. Now, it says equal there, but that is, it can be misunderstood because they are not equal in amounts. Remember, Chargaff's rule states that they are in approximate amounts, not totally equal. And they are bonded together by hydrogen bonds. So we can look here. This is adenine. Adenine is a larger double ring structure. Thymine is a single ring structure, so we can always identify them based on that. Guanine is double ringed, and cytosine is double ringed. All right, so we're looking at prokaryotic DNA, and we're going to differentiate between this DNA and the DNA of the eukaryotic. This blue picture here with the yellow chromosome shows us the circular chromosome of the bacteria. Notice that it is smaller in length. Also, bacteria have less base pairs than eukaryotic organisms. The other picture we see, the green picture with the orange bacteria, just shows that a bacterium that was broken and its DNA is leaking out. Looking at the eukaryotic DNA information, if we compare the two, prokaryotic with eukaryotic, we see that the DNA is listed on many chromosomes as opposed to just circular loop of DNA. We also know that variation exists among species for the eukaryotic DNA. Look how it's 1,000 times more than that of prokaryotes. It's also larger in diameter and length. There are also different structures that you find in eukaryotes that you don't find in prokaryotes, such as histones, nucleosomes, and chromatin. These are all different structures that help our DNA be condensed and compacted to fit on the chromosomes that we have. Now, if we're looking at this diagram, we would want to look at it and review it with our notes going from right to left, because starting on the right we have our double helix of DNA, and it's unwinding to the components that make it up. So we have the histones, and notice how the DNA is wrapped around that tightly, and then it becomes nucleosomes. We have the coils all present there, and then we see the supercoils. This is how our DNA gets wrapped around and compacted so that it fits on the chromosomes that we have. Okay, those 30,000 genes and stuff like that that we have been known to have had using the Human Genome Project as a reference, approximately 30,000 or so. DNA has to copy itself, so that's why we're going to have to go over replication, explaining to us how DNA copies itself. We already went over the basis of this, and we know that DNA copies itself in S phase of interphase. Okay, that's when the DNA is duplicated. Why? Because we want each one of our cells to have the same DNA information, and this is how it happens. So if we have one side of our DNA that has strands adenine, adenine, guanine, cytosine, thymine, thymine, and thymine, what will the new match be? So we're trying to find the complement. So that blank that exists in the notes, you have to add the complement to this strand of DNA. Prokaryotes, guys, replication in prokaryotes is very simple. You have structures like these loops here that are present. 2 new loops are made. They have a cell wall, a plasma membrane. Okay, that's not especially unique. However, the forming of these loops is. The other thing that's unique too is the fact that you have a septum forming here before the two cells separate. In eukaryotic cell division, you have a cleavage furrow forming before the cells divide. So that is a different process that happens, and a very simplified process compared to the meiotic and mitotic divisions that happen with eukaryotes. So replication in eukaryotes, as we know, is much more complicated. And again, the reason why the DNA has to replicate or duplicate or double itself is so that the same genetic information is copied and passed on to the new cells. Okay? And in this instance, the same DNA is copied so that it is passed on to the new organism. And when I mean organism, I mean new cell, new DNA, which has a template of the old strand of DNA. So, template also can be worded as a model or an example. So each new strand of DNA is a combination of the old strand and a new strand. Enzymes, very helpful structures, proteins, as we know, unzip the information, unzip the DNA molecule, allowing for the new base pairs to be added to make the new strands of DNA. It also proofreads it. Okay, so it makes sure that any kind of corrections that can be done are made. All right, so guys, number 1 represents the original strand of DNA. Number 2 illustrates when an enzyme like DNA polymerase comes along, splits the DNA in half, or unzips it is the terminology that is used. And notice those new base pairs are going to be added to the unzipped strand of DNA. So from that one original strand, now in number 3, we have 2 strands of DNA that have both old and new parts to it. This is the more realistic diagram, as it says at the top, because DNA looks like a spiral staircase or a twisted ladder in actuality. And so this shows us what it could potentially look like. It also color codes the adenines, guanines, cytosines, and thymines. Shows us that polymerase operates at more than one point, which is what actually happens. Shows us the original strand and the new strand of DNA.